deubiquitylase enzyme USP9X in head and neck cancer. This is,
if you speak into the microphone.
This is part of work that I'm carrying out for my PhD project. So, head and neck cancer is the 6th most common cancer in the world, and the cancers that arise in the oral cavity, the pharynx, larynx, and the paranasal sinuses are categorized under this head and neck cancer. There is a 5-year survival rate after diagnosis, about 50%, mainly because a late diagnosis due to the asymptomatic nature of the early tumors and to drug resistance to the currently available chemotherapies. Now, these factors emphasize the need to study the underlying molecular pathways such that we can unravel new detection markers in order to improve the diagnosis and also to discover novel drug targets to address the issue of drug resistance. Recently, a study was carried out in India on gingival buccal oral squamous cell carcinomas. Now, India is one country with the highest rate of oral cancers, attributed mainly to the habit of betel chewing that's quite popular among the locals. So this particular study characterized the somatic mutation landscape of oral carcinomas. Now, their genetic screening came up with several genes that's been already implicated in oral carcinomas, such as TP53 and HRAS. Apart from that, they also discovered 5 new genes having a link to oral squamous cell carcinomas. Now, among them was USP9X, which was found to be mutated in 22% of the patients they studied, and mostly the mutations were copy number loss or truncating mutations, which suggested that USP9X could be acting as a tumor suppressor in these oral carcinomas. Now, this study formed the basis of our current investigation. We are a lab which works very closely with USP9X, and based on these findings, we wanted to dig deeper and understand more profoundly the role of USP9X in these sort of cancers. Briefly, what is USP9X? It's a deubiquitylating enzyme, and deubiquitylating enzymes are capable of reversing the action of ubiquitination. Now, ubiquitination is a post-translational modification which occurs through an attachment of ubiquitin molecule onto a target protein. Now, this attachment of ubiquitin molecule or molecules on a target protein can decide its fate within the cell. For instance, it can result in the proteasomal degradation of the target protein. It can modify a protein such that a signaling pathway is activated, or it can even result in the localization of the protein within the cell. So, deubiquitinases are basically able to reverse all of these processes. Now, ever since the discovery of USP9X, several research has been carried out around it, and it's known to have a role in development as well as in disease. In context of cancer, several studies have been conducted in the recent past that links USP9X into cancer. To name a few: breast cancer, lymphomas, brain cancer, and lung cancer. In each, now the role of USP9X is quite tissue specific, so its role in these different cancers is quite different. So our investigation aims to evaluate the role of USP9X in head and neck cancers with the help of an in vitro system, and we also aim to elucidate the molecular mechanisms that USP9X regulate. Now, to do so, we chose 4 cell lines: SSC15, Cal27, 562. These are established head and neck carcinoma cell lines. And we found out that all 4 are expressing USP9X, so they don't have any truncating mutations in USP9X. And now there are several approaches you can take to start a protein within a cell. One such approach is to knock it down, and then study upon its depletion what are the mechanisms, cellular pathways, what are the cellular aspects that's being deregulated, such that you can get an insight into its role within a cell. So for our investigation, we chose this approach, and we used commercially available siRNAs to knock down our protein. So we found that siRNAs work quite well with our cell lines. All 4 cell lines showed a substantial decrease in our USP9X levels after 72 hours of treatment. Then we went ahead and we wanted to study the cell aspects that's been re-regulated in its absence, and we began with cell proliferation. We used cyclone assay, which is a commonly available assay to study cell proliferation. So when we treated our cells with siRNAs and monitored its proliferation, we found out that upon depletion of USP9X, the cell proliferation is decreased. It's quite evident in 3 cell lines. Cal27 showed the minimum effect. So before we move on, we wanted to find out if the differences in cell numbers that we see here is not due to an increased apoptosis. So to determine the level of apoptosis, we carried out some immunoblotting assays, and we probed for cleaved PAF1, which is a marker for apoptosis. And we found out that no elevation in apoptosis upon depletion of USP9X when compared to its controls. So which suggested that the cell number differences that we saw from cyclone assay is more to do with a reduced proliferation. So a decrease in cell numbers in the USP9X rather prompts the role of the tumor promoter. This contradicts what we expected, since an oncosuppressive role was suggested in the study I quoted earlier. However, this isn't surprising, since USP9X is known to have a context-specific or bimodal role in cancer. It can be illustrated with reference to pancreatic cancer, where in 2012, this was a study that was published in Nature, where they claimed that USP9X acts as a tumor suppressor. So when they knocked out the protein in mice that had an activated KRAS, they found that mice developed pancreatic ductal adenocarcinoma at an accelerated rate. 2 years later, when Cox et al. Tried to knock down USP9X within the established pancreatic cancer cell lines, they found this actually there is a slowdown of cancer cells when the USP9X is depleted, which suggested that it seems to be playing a tumor promoter role in cancer cell lines. So these 2 studies shows that the role of USP9X could vary from the early stage of cancer to the late stage of cancer. Now, in order to further confirm our findings with reference to cell proliferation, we switched up our approach. We overexpress USP9X now in order to see if we are seeing the same kind of effect on the cell's proliferation. Now, in order to overexpress USP9X, we used a vector known as PDS51, which has an insertion of USP9X cDNA. And we established stable cell lines. To do so, we linearized the plasmid and transfected our cells with LabFect in 2000, and we selected the transfected cells with the help of blastocycline. Now, the ectopically expressing USP9X is V5 tagged, and so we could monitor the expression of ectopic USP9X by probing for V5 tag. Then we went ahead and did our cyclone analysis once again, and quite interestingly, upon ectopic expression of USP9X, we are now seeing an increase in cell proliferation. So when we knocked it down, we saw a decrease in cell proliferation, and when we overexpress or ectopically express USP9X, we are seeing an increase. So it looks like the cell numbers are directly proportional to the level of USP9X protein, which further confirms that it seems to be playing an oncogenic role in these head and neck cancer cell lines. Now, the second aim of the study was to study the molecular mechanisms behind its regulation. Now, USP9X has several known substrates which take part in several signaling pathways. However, for our study, we narrowed it down to 3 pathways, which is Notch, mTOR, and Wnt signaling pathway, mainly because it is known to regulate the cell proliferation of head and neck cancers, and it has no substrate, USP9X substrate involved. So the activation of these pathways were monitored with the help of downstream targets, which are cyclin D1, CMYC, and HES1, and we quantitate its RNA expression by qPCRs. The experimental design was to extract RNA at 3 different time points from both siRNA treated cells as well as the cells as ectopically expressing USP9X, generate cDNA, and conduct qPCR for the target genes. So we studied the fold change expression of our target genes across all 4 cell lines at 3 different time points. This is just the data at 144 hours, which is our last time point. And we observed quite interesting patterns of expression levels in all target genes. However, the most significant correlation in expression pattern was observed with reference to HES1. Now, HES1 is a downstream target of Notch signaling pathway. 3 of the cell lines once again show a down-regulation of its expression upon knockdown of USP9X. Cat27 showed no difference. However, it's expected since we didn't really see an effect on cell proliferation when we knocked down USP9X within the cell line. When we ectopically express USP9X, we observed that the HES1 level is upregulated. So consistently, HES1 expression correlated with the cell proliferation which we measured by cyquant assay, and USP9X seems to positively regulate Notch pathway, which led to our current hypothesis, that is USP9X regulates proliferation of head and neck cancer cells through Notch pathway. We are currently conducting a few experiments to test this hypothesis. So in conclusions, we noted a decrease in cell proliferation when we knock down USP9X and an increase when we ectopically express USP9X, and it seems to positively regulate Notch pathway. Acknowledgements goes to my supervisors and my lab members, our collaborators, and the university for funding. Thank you.
Any questions for this young lady who's presented us with such a nice lecture? Yes, sir?
Just to notice that squamous cell carcinoma is not a proliferative disease. Cancer is not simply cell division. No is cyclin D1 expression cell division per se. So a lot happens in a cancer tissue, such as invasion and migration, deformation, that is not directly related to the degree of cell division. And so I'm just asking, when you said in the beginning, when you saw mutations in USP9X, your conclusion was that it could be a tumor suppressor, and then you changed over to oncogene. So 2 things. With P53, that was the case, and then it was realized that the ectopically expressed gene is actually a mutant one. So did you confirm that your ectopically expressed USP9X is wild type? And the second, yes it is. And the second is, why did you think it was tumor suppressor? The mutations were ones that, were there frame shift mutations, deletion mutations?
Yes, it was truncating mutations that was noted in that particular study.
So I fail to see how this in vitro assay convinces you that it's an oncogene. In nature, it's a tumor suppressor. That's what it is. Your experiments need to bow down to nature, not the other way around. It seems to me.
Yeah, we just emphasize the point that it could, not all cancers, or not all the oral carcinomas would have the USP9 truncating mutations, and which has the advanced form of cancers, which survive in the sense of cancers that generate without a mutation can USP9X and early stage which progresses into late stages. They could be modifying the role of USP9X.